Hey, welcome back. This is the second video about phenotyping of traits related to mature fruit of tomato, which concern its size and structure. I will share with you some tips on evaluation of fruit external color, color of the flesh, glossiness of skin, and fruit firmness. Let's begin with the next trait, which is color at maturity. The color of tomato fruits is determined by the presence of different pigments synthesized during the ripening process. A combination between chlorophyll, carotenoids, and flavonoids determines the color of ripened tomato fruits. These pigments not only give color, but they have essential role for photosynthesis, growth, and development of plants. They also protect plants for damages caused by UV radiation and attract animals for pollination and seed dispersion. When consumed, the pigments act as antioxidants to protect human and animal cells. The research also shows that tomato color relates to tomato flavor. In the CPVO technical protocol, the trait number is 37. It is asterisk trait, which means that it has to be examined and included in the variety description. It has to be assessed at full maturity state of the fruits because when unripe, all tomatoes are green. The recommended method of observation is visual assessment by a single observation of a group of plants. It is pseudo-qualitative characteristic, which means that each individual state of expression needs to be identified to adequately describe the range of the characteristic. Here, all 7 states of expression are presented. Under CPVO number column, we see a plus in parentheses. This makes a reference further in the document where it says that the trait has to be observed after a full change of the color when placenta is found clearly in the cross section. It also says that this characteristic is not applicable for genotypes homozygous for the RIN gene that do not ripen at all. Practically, observations are made on harvested mature fruits from second or third truss of at least 20 plants. In our Harnestone phenotyping trial, we are going to assess this trait both qualitatively and quantitatively. For qualitative evaluation, we will use the CPVO scale with one change in the last state of the descriptor. Instead of green, we put other, which will cover all other colors. For quantitative evaluation, we will use International Commission on Illumination color space. This color space expresses color as 3 values, L for lightness, A and B for the 4 unique colors of human vision, red, green, blue, and yellow. These parameters will be measured by chromometer at the full ripe state of the midpoint of the fruit length. We are going to do 2 opposite measurements per fruit. Let's move to the next trait, which is color of flesh. The color of flesh is not necessarily the same as the color of fruit. For example, pink fruits usually have red flesh, since external pink color is due to the colorless and transparent skin. In the CPVO technical protocol, the trait number is 38. Similar to the fruit color, flesh color is asterisk trait that has to be included in the variety description. It is pseudo-qualitative characteristic with 7 states of expression. It has to be assessed at full maturity state of the fruits. The recommended method of observation is visual assessment by a single observation of a group of plants or parts of plants. Practically, observations are made on harvested mature fruits from second or third truss from at least 20 plants. In our Harnestone phenotyping trial, we are going to assess this trait by image analysis using Tomato Analyzer software, but before using it, we have to scan vertically cut fruits at high resolution of 300 dots per inch. We will continue with the trait number 39, which is glossiness of skin. Skin glossiness is important for visual appeal and marketability. Tomatoes with shiny, smooth, and blemish-free skin are generally more visually attractive and may be more appealing to the consumers. In some markets, glossy tomatoes may also be perceived as fresher and of high quality. It is quantitative characteristic. The recommended method of observation is visual assessment by a single observation of a group of plants that has to be done on fully ripened fruits. Here, the trait has only three expressions: weak, medium, and strong. Practically, observations are made on harvested mature fruits from second or third truss from at least 20 plants. The skin of tomatoes should be dry and clean, and before evaluation, tomatoes should not be refrigerated since the cold temperature may cause a dour appearance. The examination has to be made under bright light or natural light. A glossy tomato will reflect light and will have a shiny appearance. In our Harnestone phenotyping trial, we are not going to assess this trait. And the last trait is fruit firmness. Fruit firmness is a measure of a texture of tomato fruits and refers to the degree of hardness or softness of the fruit. It affects the shelf life, transportability, and consumer acceptance of the fruit. This is trait number 40 in CPVO protocol and 41 in UPOV. CPVO technical protocol tells that this trait is important for the variety description. It is a quantitative characteristic. The recommended method of observation is visual assessment by a single observation of a group of plants. Here we have 9 states of expression given in an abbreviated scale 1, 3, 5, 7, and 9. But all 9 states of expression should be used to describe the variety. So, between very soft and soft state, we have very soft to soft, soft to medium, medium to firm, and firm to very firm. Practically, observations are made on harvested mature fruits of second or third truss from at least 20 plants. To determine the average image, leave aside the largest and the smallest fruits in the sample. Also leave aside the damaged fruits. Determine the firmness of the fruit by gently squeezing it and compare with the example varieties to decide on the proper state. In our Harnestone phenotyping trial, we are going to assess this trait in more quantitative way by using a durometer. This is a device that applies a controlled force to the fruit and measures the resulting deformation or resistance. Firmness will be measured at the midpoint of the fruit length in two opposite points of the fruit. Now it's time to thank you for watching these videos. Thank you for investing your time in learning. I highly appreciate your engagement. Please ask questions in the chat or leave your comments. Your feedback is valuable for us and will help us to improve future content. Thank you.